Hello students, how are you all? I hope you are doing well. I am here to teach you chapter 2 microorganisms. Let's warm up. Look at this picture. Have you ever seen a rotten slice of bread? What do you think might have caused the bread slice to rot? So students, here look at this picture and you have to write some lines about it. Let's know about microorganisms. Discovery of microorganisms, general characteristic of organisms, classification of organisms. Here we have microorganisms. The presence of moisture allows certain microorganisms to grow in leftover food which cause it to rot. These tiny microorganisms are called microorganisms or microbes. So student, जो moisture की presence में grow करते हैं, that is microorganisms, or we can say that microbes. Discovery of microorganisms. The discovery of microorganisms is closely related to the advancement of microscope. We could know all about microorganisms only after the development of microscope. Anton van Leeuwenhoek in 1657 discovered very small living creatures in rainwater using his newly constructed microscope. Here, Louis Pasteur, the father of bacteriology, discovered microorganisms in the air and concluded that they are responsible for fermentation of food and disease in man and animals. He gave the famous theory, the germ theory of disease. Robert Koch designed severe techniques of handling bacteria. Here we have general characteristics of microorganisms. Microorganisms are a collection of undifferentiated cells. They are visible only under microscope. They are found in air, water and soil and on the body of living organisms. They can survive in extreme conditions of temperature and rot in hot water, springs as well as marshy lands. Do you know? The study of microorganisms is called microbiology. Here we have an activity, activity 1. Collect some moist soil from the field in a beaker and add water to it. After the soil's particles have settled down, observe a drop of water from the beaker under a microscope. What do you see? So student, you can perform this activity with the help of your teacher. Here we have activity 2. Take a few drops of water from a pond, spread on a glass slide and observe through a microscope. Now we have bacteria. Bacteria are most abundant of all the microorganisms. They are simplest of living organisms. Here, habitat. Bacteria are most abundant of all the microorganisms. They are simplest of living organisms. Here, habitat. Bacteria occur almost everywhere on the earth, in the air, in the water, in the sky, and in food product and also in the body of other organisms. So bacteria आपको बच्चों हर जगह देखने को मिलते हैं. Now we have size. The size of bacteria varies from 0.2 to 100 microns. 1 micron is equals to 1,000 of a millimeter. Here, shape. On the basis of shapes, bacteria are classified into the following types. Here, cocci, singular coccus, spherical bacteria, bacilli, singular bacillus, rod-shaped bacteria, spirulia, singular spirillum, spiral bacteria, vibrio, coma-shaped bacteria. Now, students, look at these pictures. These are the different shape of bacteria. These are the bacteria of different shapes. Now we have check and mate, as we already read about these things, so you can do it by yourself. Now we have fungi. Fungi are plant-like non-green multicellular organisms. Some of them look like plants but do not have roots, stem, leaves or flowers. So students, fungi plants की तरह दिखाई देता है लेकिन होता नहीं है. उसकी कोई stem नहीं होती, root नहीं होती, कोई flowers भी नहीं होते हैं. Some fungi like yeast are unicellular while others like mushrooms, penicillium, toads, stools, sponge fungi, molds, and mildews are multicellular. Fungi are non-green plants and hence they cannot make their food. They are either saprophyte or parasites. So students, fungi क्या होते हैं? Non-green plants होते हैं, तो वो अपना खाना खुद नहीं बनाते हैं, या तो वो saprophyte होते हैं या फिर parasites होते हैं. Fungi reproduce asexually by budding, yeast, or by forming dust-like spores in mushrooms and molds. Spores remain in the air and help in the dispersal of fungi to distant places. So students, fungi जो है वो reproduce करती है asexually by budding, yeast or forming dust-like spores. Here we have some pictures. Look at the picture. We have yeast, sponge fungi, then penicillium, then mushrooms, some common fungi. Now we have algae. Have you ever seen a layer of green scum growing in stagnant water? It is actually algae growing on the surface of water. So student, algae कहाँ grow करती है? Surface of water. Algae are aquatic organisms. They are plant-like in their ability to photosynthesis. However, they do not have true roots, a stem, and leaves. They are found in fresh water, wastewater, and sea. Most algae are microscopic, but some, like seaweeds, are quite large. Some algae, like Climodomerus and Chlorella, are unicellular, whereas others like kelp and Spirogyra are multicellular. They are given red and brown algae depending on the type of pigments present in the air. So students, हमारे पास red और brown algae भी होते हैं, वो depend करता है कि pigment कैसा present है. Now look here, we have Climodomerus nostoc. These are the algae. Here, protozoa. Protozoa are the most primitive single-celled microscopic animals. Amoeba, Paramecium, Euglena, Antiamoeba, and Plasmodium are some common protozoans. Most protozoans occur single, but some are colonial also. They do not possess cell wall as in plant cells. Their body is either with plasma membrane alone or is covered with palisade. They can swim using either flagella or cilia or can move with the help of pseudopodia. So student, इनमें plant cell की तरह cell wall present नहीं होता है और ये मूव करते हैं या तो स्यूडोपोडिया की हेल्प से या फिर प्लाज्मा की हेल्प से भी स्विम भी कर सकते हैं. नाउ लुक एट द पिक्चर्स. वी हैव पैरामीशियम, अमीबा, यूग्लेना, सम कॉमन प्रोटोजोआ. नाउ वी हैव वायरसेस. सो स्टूडेंट, वैसे तो आप सबने सुना होगा वायरस के बारे में. नाउ रीड अबाउट इट. Viruses are microscopic organisms much smaller than bacteria. You might be familiar with viral fever, chickenpox, measles, and conjunctivitis. These are diseases caused by viruses. Viruses are vesicular organisms which exist in two states. When outside a living body or cell, they are like non-living things and do not show any feature of a living thing. They are unable to grow and reproduce outside a living cell. Viruses in their non-living state are called virions. However, when they enter a living cell, they start reproducing like other living organisms. They need a living cell in order to grow. They are therefore considered to be on the borderline of living and non-living. All viruses are parasitic. Look at the picture, we have virus. So student, अगर हम viruses की बात करें, जैसे अभी आप लोग इस फेज से बाहर आए हैं, हम ओवरकम हुए हैं कोरोना वायरस. तो कोरोना वायरस क्या था? वायरस था. कोविड हुआ किसकी वजह से? वायरस की वजह से. सो स्टूडेंट, वायरस क्या है? एक बॉर्डर लाइन है लिविंग और नॉन लिविंग के बीच में. कैसे? जब वो किसी सेल या किसी बॉडी से बाहर होता है तो वो एक नॉन लिविंग थिंग की तरह रिएक्ट करता है, बट जब वो किसी बॉडी या किसी सेल में एंटर करता है तो वो रिप्रोड्यूस करता है और माइक्रो ऑर्गेनिजम्स की तरह और एक लिविंग थिंग की तरह बिहेव करता है. गेट इट? इज दैट मीन्स उसको रिप्रोड्यूस या फिर ग्रो करने के लिए किसी लिविंग सेल की नीड होती है. नाउ वी हैव friendly microorganisms. Microorganisms are used for various purposes. They are used in the preparation of curd, bread, and cake. Microorganisms have been used for the production of curd, bread, and cake. They are also used in cleaning up of the environment. For example, the organic wastes, vegetable peels, remains of animals, feces, etc. are broken down into harmless and usable substances by bacteria. Recall that bacteria are also used in the preparation of medicines. In agriculture, they are used to increase soil fertility by fixing nitrogen. So अगर स्टूडेंट्स हम फ्रेंडली माइक्रोऑर्गेनिजम्स की बात करें, जैसे आपने अभी पढ़ा कि वो कर्ड की फॉर्मेशन में हेल्प करते हैं, ब्रेड की फॉर्मेशन में हेल्प करते हैं, और भी बहुत सारी चीजें हैं जिसमें वो हेल्प करते हैं, जैसे प्रिपरेशन ऑफ मेडिसिन में हेल्प करते हैं. अगर हम एग्रीकल्चर में बात करें तो नाइट्रोजन फिक्सिंग में हेल्प करते हैं जो सॉइल की फर्टिलिटी को इंक्रीज करते हैं. नाउ वी हैव मेकिंग ऑफ कर्ड एंड ब्रेड. Milk is turned into curd by bacteria. Curd contains several microorganisms. One of these, the bacterium Lactobacillus, promotes the formation of curd. It multiplies in milk and converts it into curd. Bacteria are also involved in the making of cheese, pickles, and many other food items. An important ingredient of rava, suji, idlis, and bhaturas is curd. Can you guess why? Bacteria and yeast are also helpful for fermentation of rice idlis and dosa batter. So students, अगर हम बात करते हैं ये क्या होता है, हम भटूरे के आटे में क्या, जब हम डो बनाते हैं भटूरे के लिए कर्ड ऐड करते हैं या फिर यीस्ट पाउडर ऐड करते हैं. क्यों करते हैं? क्योंकि हमें वहां फर्मेंटेशन करानी होती है, जो किसकी वजह से होती है? बैक्टीरियाज की वजह से होती है. नाउ वी हैव एन एक्टिविटी 3. Take half kg flour, atta or maida, add some sugar and mix with warm water. Add a small amount of yeast powder and knead to make a soft dough. What do you observe after 2 hours? Did you find the dough rising? Look at the picture, student. We have maida with yeast powder, raised maida. Yeast reproduces rapidly and produces carbon dioxide during respiration. Bubbles of the gas fill the dough and increase its volume. This is the basis of the use of yeast in the baking industry for making breads, pastries, and cakes. Now we have commercial use of microorganisms. Microorganisms are used for the large-scale production of alcohol, wine, and acetic acid vinegar. Yeast is used for commercial production of alcohol and wine. For this purpose, yeast is grown on natural sugars present in grains like barley, wheat, rice, crushed fruit juices, etc. So student, हमारे जो माइक्रो ऑर्गेनिज्म है उनका बहुत बड़ा रोल होता है अल्कोहल इंडस्ट्री में. बहुत ज्यादा यूज किया जाता है. अल्कोहल की फॉर्मेशन किसकी वजह से होती है? yeast की वजह से होती है. Now look here students, we have an activity 4. Take a 500 ml beaker filled up to 3/4 with water. Dissolve 2 to 3 teaspoons of sugar in it. Add half a spoon of yeast powder to the sugar solution. Keep it covered in a warm place for 4 to 5 hours. Now smell the solution. Can you get a smell? This is the smell of alcohol, as sugar has been converted into alcohol by yeast. This process of conversion of sugar into alcohol is known as fermentation. So students, alcohol किस process से बनता है? That is fermentation. Louis Pasteur discovered fermentation in 1857. Medicinal use of microorganisms. Whenever you fall ill, the doctor may give you some antibiotics, tablets, capsules, or injections such as penicillin. The source of these medicines kill or stop the growth of disease-causing microorganisms. Such medicines are called antibiotics. These days, a number of antibiotics are being produced from bacteria and fungi. Streptomycin, letracycline, and anthromycin are some of the commonly known antibiotics. Streptomycin, tetracycline, and erythromycin are some of commonly known antibiotics which are made from fungi and bacteria. The antibiotics are manufactured by growing specific microorganisms and are used to cure a variety of disease. So students, अब हम बात करते हैं कि मेडिकल फील्ड में क्या यूज है. आप सब एंटीबायोटिक्स खाते हैं, आप सबको एंटीबायोटिक्स के बारे में पता है. वो बनती किसकी हेल्प से है? हमारे माइक्रो ऑर्गेनिजम्स की हेल्प से. करते क्या है? अंदर जो डिजीज कॉजिंग माइक्रो ऑर्गेनिज्म है वो उनको किल करती हैं. So student, that's how आपके माइक्रो ऑर्गेनिज्म हमारी मेडिकल इंडस्ट्री में भी हेल्प करते हैं. Now we have in 1929 Alexander Fleming was working on a culture of disease causing bacteria. Suddenly he found the spores of a little green mold in his culture plates. He observed that the presence of mold prevented the growth of bacteria. In fact, it has also killed many of these bacteria. From this, the mold penicillin was prepared. So student, इस process की help से क्या prepare किया गया था? That is penicillin. Get it? Now we have vaccine. When a disease-carrying microbe enters our body, the body produces antibodies to fight the invader. The body also remembers how to fight the microbe if it enters again. If dead or weakened microbes are introduced into a healthy body, the body fights and kills the invading bacteria by producing suitable antibodies. The antibodies remains in the body and we are protected from the disease causing microbes forever. This is how a vaccine works. Several diseases, including cholera, tuberculosis, smallpox and hepatitis can be prevented by vaccination. So student, अगर हम वैक्सीनेशन की बात करें, देखो हमारी बॉडी क्या है, उसको आदत है हमारी बॉडी में कोई भी ऐसा वायरस अगर एंटर करता है तो बॉडी क्या करती है? उससे फाइट करती है. बॉडी में एंटीबॉडीज बनती हैं. अब कभी कभी वो एंटीबॉडीज कम होती हैं, तो उन एंटीबॉडीज के बने रहने के लिए, उन्हें अच्छी पावरफुल एंटीबॉडीज बनाने के लिए हमें वैक्सीन्स लगती हैं. और वो वैक्सीन्स जो है वो उन एंटीबॉडीज को पावरफुल बना देती हैं, ताकि अगर ऐसा कोई माइक्रोब हमारे अंदर जा भी रहा है तो हमें वो बीमारी ना हो. Here we have Edward Jenner discovered the vaccine for smallpox in 1798. In your childhood, you must have been given injections to protect yourself against several diseases. Can you prepare a list of these diseases? You may take help from your parents. So students, आप ये बहुत easy thing आप कर सकते हैं. आप अपने मम्मी पापा से पूछें बचपन में आपको कितनी वैक्सीन लगी हैं, कौन कौन सी वैक्सीन लगी हैं, और आप उसकी एक टेबल या एक चार्ट प्रिपेयर कर सकते हैं. It is essential to protect all children against these diseases Necessary vaccines are available in the nearby hospitals. You might have seen the advertisement on TV and newspapers regarding protection of children against polio under the Pulse Polio program. Polio drops given to children are actually a vaccine. So students, आप सबको पता है पोलियो के बारे में, तो पोलियो भी एक वैक्सीन है और ऐसी बहुत सारी चीजें हैं और बताया जाता है, लोगों को अवेयर किया जाता है अबाउट द वैक्सीन. A worldwide campaign against smallpox has finally led to its eradication from most parts of the world. These days, vaccines are made on a large scale from microorganisms to protect humans and other animals from several diseases. So, student, आज के टाइम में जो वैक्सीन्स बनती हैं वो माइक्रो ऑर्गेनिज्म से बनाई जाती हैं ताकि एनिमल्स और ह्यूमंस को प्रोटेक्ट किया जा सके बहुत सारे डिजीज कॉजिंग माइक्रो ऑर्गेनिजम्स इन ह्यूमन. पैथोजेंस एंटर आवर बॉडी थ्रू द एयर वी ब्रीथ, द वाटर वी ड्रिंक और द फूड वी ईट. They can also get transmitted by direct contact with an infected person or carried by an animal. Microbial disease that can spread from an infected person to a healthy person through air, water, food or physical contact are called communicable disease. Example of such disease include cholera, common cold, chickenpox and tuberculosis. So students, अब हम बात करते हैं कि आप सबको पता है कुछ डिजीज ऐसी होती है जो फैल जाती है. फैलती कैसे? या तो वो फिजिकल टच से फैलेंगी या उनका जो बैक्टीरिया या जो भी उनका माइक्रो ऑर्गेनिज्म है जो उस बीमारी को बढ़ाता है वो आपके अंदर जाएगा बाय एयर, बाय वाटर, व्हाटएवर इट इज. तो दीज आर द कम्युनिकेबल डिजीज. जो बीमारी किसी के छूने से या फिजिकल कॉन्टेक्ट से फैलती है दैट इज कम्युनिकेबल डिजीज, या फिर उनके इन्फ्लुएंस में आने से भी ये डिजीज हो जाती है, दैट इज कोलेरा है, कॉमन कोल्ड है, चिकन पॉक्स है, ट्यूबरक्लोसिस है. तभी आप लोगों को बोला जाता है कि अगर आपको कॉमन कोल्ड या स्नीजिंग या आप कफिंग करते हैं तो फेस को, माउथ को कवर करके करें. Now, when a person suffering from common cold sneezes, fine droplet of moisture carrying thousands of viruses are spread in the air. The virus may enter the body of a healthy person while breathing and cause infection. Get it, students? Now, there are some insects and animals which act as carriers of disease causing microbes. Housefly in one such carrier. The flies sit on the garbage and animals excreta. Pathogens stick to their bodies. When these flies sit on uncovered food, they may transfer the pathogens. Whoever eats the contaminated food is likely to get sick. So it is advisable to always keep food covered. Avoid consuming uncovered items of food. Another example of a carrier is the female Anopheles mosquito, which carries the parasite of malaria, Plasmodium. Female of dengue mosquito act as carrier of dengue virus. How can we control the spread of malaria or dengue? So स्टूडेंट, आप सबको पता है डेंगू और मलेरिया किसकी वजह से होता है? मच्छर के काटने की वजह से होता है. बट उनके खुद के वायरसेस होते हैं जो हमारी बॉडी में किसके थ्रू किए जाते हैं, किसके थ्रू एंटर करते हैं? मॉस्किटो की बाइट की वजह से. All mosquitoes breed in water. Hence, one should not let water collect anywhere in coolers, tires, flower pot, etc. By keeping the surroundings clean and dry, we can prevent mosquitoes from breeding. Try to make a list of measures which help to avoid the spread of malaria. So student, हमें मलेरिया और डेंगू को अवॉइड करने के लिए अपने आसपास सफाई रखनी चाहिए, पानी इकट्ठा नहीं होना चाहिए, बिकॉज जो हमारे मॉस्किटोज हैं वो ब्रीड कहाँ करते हैं? वाटर में करते हैं. Some common human disease caused by microorganisms. Here, human disease, tuberculosis, positive microorganism, bacteria, mode of transmission, air, preventive measures, general. So students, look here. Measles virus, air. Chickenpox virus, air contact. Polio virus, air and water. And now we are on preventive measures. Keep the patient in complete isolation. Keep the personal belongings of the patient away from those of the others. Vaccination to be given at suitable age. Now some more disease we have. Cholera, typhoid, bacteria, water, food, water. And here is preventive measures. Maintain personal hygiene and good sanitary habits. Consume properly cooked food and boiled drinking water and vaccination. Now come to the next, we have hepatitis A, virus, water. Drink boiled drinking water, vaccination. Malaria, protozoa, mosquito. Use mosquito net and repellents, spray insecticides and control breeding of mosquitoes by not allowing water to collect in the surroundings. Some of the common diseases affecting humans, their mode of transmission and few general methods of prevention are shown in the table. Now we have disease causing microorganisms in animals. Several microorganisms not only cause disease in humans and plants, but also in other animals. For example, anthrax is a dangerous human and cattle disease caused by a bacterium. Foot and mouth disease of cattle is caused by a virus. Look here, Robert Koch 1876 discovered the bacterium Bacillus anthracis which cause anthrax disease. Now, disease causing microorganisms in plants. Several microorganisms cause disease in plants like wheat, rice, potato, sugarcane, orange, apple and others. The disease reduce the yield of crops. See above the table for some such plant disease. They can be controlled by the use of certain chemicals which kill the microbes. So student, microorganisms जो है वो disease create animals में भी करते हैं, plants में भी करते हैं, humans में भी करते हैं. Now look at the table. We have some common plant disease caused by microorganisms. Here plant disease, microorganism, mode of transfer. Citrus canker, bacteria, air. Rust of wheat, fungi, air seed. Yellow vein, mosaic of bhindi, okera, virus, insect. Now we have food poisoning. Robbie was invited by his friends to a party and he ate a variety of food stuff. On reaching home, he started vomiting and had to be taken to a hospital. The doctor said that this condition could be due to food poisoning. So students, वैसे ये common है, आप सबने सुना होगा food poisoning के बारे में. Let's read about it. Food poisoning could be due to consumption of food spoiled by some microorganisms. Microorganisms that grow on our food sometimes produce toxic substances. These make the food poisonous, causing serious illness and even death. So it is very important that we preserve food to prevent it from being spoiled. So students, जो खाना हम खाते हैं उसमें जो microorganisms होते हैं, कई बार वो खाने में toxic substances को produce कर देते हैं, जिसकी वजह से खाना पॉइजनस हो जाता है और उसके खाने से हमारी डेथ भी हो सकती है. सो दैट्स व्हाई हमें खाने को बहुत अच्छे से प्रिजर्व करना चाहिए. नाउ वी हैव फूड प्रिजर्वेशन. Microorganisms spoil our food. Some bacteria and fungi grow over them when left unused for some days. You know, a bread slice left for 2 to 3 days under moist conditions gets covered with wild, cottony, fluffy mass. It is a fungus that means mucor. The wastage of food can be reduced by proper methods of food preservation. So student, अगर हम food preservation की बात कर रहे हैं, अगर आप खाने को ऐसे खुला छोड़ देते हैं वो खराब हो जाता है, तो हमें उसे एक proper तरीके से preserve करना चाहिए. Methods of food preservation. Different food items need different methods of preservation. Some of these methods are. So student, different type के food को क्या चाहिए? Different type का preservation. Obvious है. Now we have sun drying. The methods is used for preserving and storing raw vegetables and certain fruits. Drying in the sun reduces the moisture content of food material. As microorganisms do not grow in dry conditions, it becomes easy to store dried vegetables like cauliflower, fenugreek and mint, and fruits like mango, apple and peach in all right containers, in airtight containers. सो स्टूडेंट, सन ड्राई एक मेथड है. जैसे हमारी वेजिटेबल्स या कुछ भी चीज है उसको अगर हम धूप में सुखा के रखते हैं तो उसमें माइक्रो ऑर्गेनिजम्स के होने की चांसेस बहुत कम हो जाते हैं. नाउ वी हैव प्रिजर्वेशन बाय कॉमन सॉल्ट. प्रिजर्वेशन ऑफ फूड बाय एडिंग सॉल्ट इज कॉल्ड सॉल्टिंग. द मीट एंड फिश आर कवर्ड विद सॉल्ट टू ड्रेन आउट वाटर बाय ऑस्मोसिस. Raw mango, lemon, amla, tamarind and other fruits and vegetables are preserved by salting. This inhibits growth of bacteria. Preservation by sugar. Sugar syrup containing 68% sugar is used for the preservation of fruits and vegetables. So student, sugar भी use किया जाता है, salt भी use किया जाता है preservation के लिए. Such a high concentration of sugar reduces moisture content and thus prevent growth of microbes. Carrots, plums, ripe mangoes, amla, apples, etc. are preserved by sugar in the form of jams, jellies and squeezed. So इस तरीके से हम sugar की help से भी preserve करते हैं. Now, preservation by pickling. Oil and vinegar are used to make pickles because bacteria cannot survive in high acid contents of the medium. Vegetable fruits like raw mangoes, lemon, chilies, etc. are preserved as pickle. Salt or sugar is added to the pickle to enhance its taste. Now, prevention by chemicals. Citric acid is added to pickles and some squeezes, while sodium benzonate and sodium metabisulfite are used as preservation to preserve squeezes, jams and jellies. So student, ये कुछ chemicals हैं जो हम preservation के लिए use करते हैं. Now, preservation by pasteurization. Pasteurization is the process of heat and cold treatment to which milk is subjected to make it bacteria free. The milk is heated to 62 degrees Celsius for 30 minutes or 70 degrees Celsius for about 15 to 30 seconds and then chilled suddenly. The process of pasteurization was discovered by Louis Pasteur. The milk treated this way is called pasteurized milk and can be safely used without boiling. So student, इस method से हम milk को preserve करते हैं. Now, preservation by deep freezing. Deep freezing prevents bacterial growth and inactivates the enzymes. Therefore, deep frozen food retains flavor as well as freshness. So हम deep freezing से भी preservation करते हैं. Now students, what we learn in this chapter, we are going to discuss it in a nutshell. The organisms which we cannot see in the air naked eyes are called microorganisms. Anton van Leeuwenhoek in 1657 discovered very small living creatures in rainwater using the newly constructed microscope. Microorganisms are found practically everywhere and in all types of environments. Bacteria are most accident of all the microorganisms. Fungi are plants like non-green multicellular organisms. Algae are simple unicellular or multicellular green plants. Protozoa are the most primitive single-celled microscopic animals. Here we have some keywords. Microorganism, very small organism to be seen with naked eye. Bacteria, one-celled microscopic living organisms. Fungi, plant-like non-green multicellular green plants. Algae, simple unicellular or multicellular green plant. Protozoa, the most primitive single-celled microscopic animals. Viruses, the smallest and most primitive cellular microorganisms. Thank you students. We will meet again in the next chapter. Till then, revise what we learned today.